A little more than 10 years ago, Thomas Angelini and colleagues were studying the motion of a layer of cells growing on a petri dish. These cells were confluent, that is, there are no gaps between them. The scientists monitored the motion of the cells and noted how dense the layer became over the range of time. While recording these observations, they noticed a peculiar kind of dynamics, something very similar to what observed in glass. But glass molecules, movement, what does all of this mean? Before going deeper into this observation, let me tell you about another study. A few years down the line, another group of researchers recorded the movement of epithelial cells in culture. So over time, as the layer became denser, the movement of the cells slowed down. This reminded the scientists of how the movement of the glass molecules slow down when a liquid is quickly cooled down to like solid-like form. These studies reported a similarity between a dense layer of moving cells and solid-like glassy system. However, there was no theoretical explanation to what was going on. Is there a unified theory that can explain this behavior? I started working with Soros in the middle of 2019. Before this, from the courses that are offered in the institute, I got a taste of ongoing research on theoretical physics. This motivated me to join him since he works on these aspects. So I went to him and talked about my research interest. From time to time, we kept on discussing many problems. Initially, I had some hesitation about working on biological application-based problem because I belong to a core physics background. But Soros told me to have a look at some papers about glass and glassiness in biological tissue. In the beginning, I was surprised to learn about some of the experimental evidence showing glass-like signature in dense biological tissue. Now, not all cells are glass-like. When they become cancerous or asthmatic, they show glass-like properties. I got interested in this topic and started building up an analytical theory along with a computer simulation on a model of dense tissues. And that's how it all started. So glass is a very mysterious material. Although it looks solid to the bare eyes, its microscopic structure resembles liquid. It has been seen in simulation that over long time scale, this long can be of the order of age of universe. And we'll see that the glass indeed flows like a liquid. These strange properties of glass make it one of the deepest unsolved problems of physics. Most solids have an order structure of molecules quite similar to the neatly stacked oranges in this fruit cart. These are crystalline solids. On the other hand, liquids have an arrangement of molecules that resemble the random arrangements of oranges, as you can see here. When glass is in liquid form, its arrangement is similar to this. When it quickly cools down to a solid, this arrangement is still random and your window glass is formed. All this while, we saw studies that reported a glass-like behavior in confluent cell layers, but there is no theory that explains these observations. So Soros and I decided to come up with a theoretical explanation of such dynamics in living cells. So basically we had three major steps to cover. Develop a simulation that could capture the glass-like dynamics in a layer of cells, come up with a unified theory, and tally existing experimental data with simulation.